Welcome students to another exciting lecture. Today's topic is action potentials. And I want to encourage all of you who are starting to look at this lecture that if you haven't looked at my lecture on resting membrane potentials, to pause now and to go and review that lecture, because the generation of the resting membrane potential provides the basis for us understanding action potentials and how they're generated in the body. So let's begin. We want to begin by giving a basic definition of what is an action potential. So an action potential is a transient and regenerative change in the membrane potential that transmits electrical signals along nerve fibers. So you can see in this diagram here a neuron, and you can see these little balls of light here. These represent action potentials. Now, action potentials get generated close to the cell body or cell soma, often in a region called the axon hillock. And from here, they travel down the entire length of the axon to the terminal end of the axon, where they can communicate with other nerve cells or other parts of the body. And therefore, the real purpose of an action potential is it allows communication within the body between nerve cells and between the nervous system and other parts of the body. Note that we said the action potential is transient. It's not like the resting membrane potential that is there all the time. And it's also regenerative. And in a little while, we'll explain what we mean by regenerative. Now, before we go much further, we just want to talk about a difference between two types of potentials: action potentials and local potentials. In this diagram, we have a series of two nerve cells, nerve A and nerve B. Nerve A is going to communicate with nerve B. And to achieve that, an action potential is generated in nerve A, which travels along its length towards its end. But in order to communicate between nerve A and nerve B at the synapse, there's a release of a neurotransmitter. This carries the signal across and generates a local potential here within the cell body of nerve B. If the local potential is big enough, it will generate another action potential in cell B. And so here we can see that nerves can generate both action potentials and local potentials. What's the difference between the two? Well, a local potential is usually a much smaller change in the resting membrane potential. And local potentials cannot be propagated, or they cannot travel over long distances. That's why they're called local potentials. They stay very close to their point of origin. And as they begin to move away from their point of origin, they get much smaller and eventually disappear. Local potentials don't have the potential or the ability to regenerate. And therefore, because of this, they are not action potentials. They are not regenerative. Now, to help us understand this a little bit more, let's look at what is a very common setup in an electrophysiology laboratory. So very often in an electrophysiology laboratory, we have a nerve cell. And the nerve cell will have a recording electrode, which is connected to an amplifier. And it will have a stimulating electrode. And you can change the size of the current applied by the stimulating electrode, and then observe what happens to the potential or the change in the resting membrane potential by measuring it on the recording electrode. Now this graph here on the bottom right of your screen, that tells you the particular stimulus that we're applying via the current electrode. And then the graph on the top right shows you the change in the resting membrane potential. And you should notice some features. We can apply currents that are both negative and positive. And we can also apply currents of different sizes. So notice current 1 is more negative than current 2. And current 4 is more positive than current 3. So we have the ability to supply or stimulate the nerve with different types of currents according to its polarity or charge and according to its size. And what you notice is, depending on the current that we stimulate with, we get a different response in the nerve. If we stimulate with a negative current, the resting membrane potential becomes more negative. If we stimulate with a positive current, the resting membrane potential becomes more positive. Now, these movements in different directions have particular names. When you move in a more negative direction away from the resting membrane potential, as is seen in the diagram on the bottom left, a more negative direction, we call that hyperpolarization. When you move in a more positive direction, you call that a depolarization. So we can see that these small currents, these localized potentials generated here, they can be both depolarizing, as is seen in example 3, but they can also be hyperpolarizing, as seen in examples 1 and 2. Example 4 is actually an action potential. And one of the things you notice is an action potential only moves in one direction, up. Action potentials are always depolarizing. The other thing you should notice about these local potentials is, depending upon the size of the current, it changes the size of the local potential. So if we give a bigger negative current, as in 1, you get a bigger response, as compared to 2, which is a much smaller response. And so we can say that local potentials are graded. They can have different sizes. Action potentials, on the other hand, are always the same size. And we're going to explain why that comes about in a little while. So let's put together a little chart that allows us to compare local potentials and action potentials. Now, some of these features of the action potentials you might not fully understand, but they'll be explained as we go through the lecture. So we saw with local potentials that you can get a graded response. That means the response can be of different sizes. However, action potentials are all or nothing. Either you get an action potential or you do not get an action potential. And when you get it, it will always be the same size. Local potentials can be added together. That means you can have two local potentials and they can come together to create a different sized local potentials. They can be summated. That tends to happen at the synapse. Action potentials, because they're all the same size for a particular nerve, they cannot be summated. We just saw that local potentials can either be depolarizing or hyperpolarizing, whereas action potentials can only be depolarizing. And finally, based on the definition of local potentials, they are conducted with decrement, or they decrease in size as they move from their point of origin, whereas action potentials are regenerative, so they are conducted without decrement. They don't change in size as they move away from their point of origin. You will study local potentials more if you study the synapse, where you have your classic example of a local potential that is generated. Now let's talk about the action potential in more detail. And you can see an action potential diagrammed on the left of your screen. Here you see that an action potential represents a change in membrane potential with time. So anytime you have to draw a diagram of an action potential, it's very, very important that you include your axes. If you don't include your axes, you're not really drawing an action potential. Note the units on the axes. On the y-axis, we have the membrane potential measured in millivolts. And on the x-axis, we have time measured in milliseconds. Now, all action potentials have 4 phases. Phase 0 is this region here, which corresponds to the resting membrane potential and any small local potentials that are generated by membrane depolarization. It's the initial slow depolarization that sometimes occurs when you stimulate a cell. Phase 1 has several other names. Sometimes it's called the upstroke or the rising phase or the depolarizing phase. And that's why I prefer to use the term phase 1. That nomenclature is very clear about what phase we're talking about. Phase 2 is this downward slope here, often called the downstroke or the repolarization phase. Phase 3 is this small region here where the membrane potential becomes actually more negative than the resting membrane potential. It's often called the overshoot or the hyperpolarization phase. And then you notice the RMP returns back to normal, and we're back here in phase 0. So those are the different phases of the action potential. And what we want to do now is we want to look at what are the ionic changes that are responsible for each component of the action potential. So let's do that by focusing now on phase 0 and how it transitions to phase 1. Now phase 0 here is the resting membrane potential. And the resting membrane potential exists because there's a greater movement of potassium out of the cell due to the permeable leaky channels than there is of sodium into the cell. Now, if you were to apply a current to the cell, what happens is that current opens a population of voltage-gated sodium channels. Now, you remember that we spoke about the different types of ion channels that exist. And one population of ion channels are voltage-gated channels. These open in response to a change in membrane potential. So when you apply a current that depolarizes the membrane, it causes the voltage-gated sodium channel to open. Now, when the voltage-gated sodium channel opens, what do you think is going to happen? Well, you will remember that the concentration of sodium on the outside of the cell is higher than the concentration of sodium on the inside of the cell. So if a voltage-gated sodium channel opens, that means we've increased the permeability of the cell membrane to sodium, and sodium is going to enter the cell, flowing through these voltage-gated sodium channels. So that's the first thing that I need you to understand, and I need you to appreciate, that when we open voltage-gated sodium channels, sodium is going to move down its concentration gradient, and it's going to flow into the cell. Now, when sodium flows into the cell, what we're actually doing is we're bringing positive charge into the cell. And we just saw that when you bring positive charge into the cell, the membrane potential becomes more positive. And if the membrane potential becomes more positive, what do we call that? We call that a depolarization. Now, track with me very carefully. When the cell membrane depolarizes, what happens to the voltage-gated sodium channels? Remember, they open. Now, when they open, what happens to sodium? Sodium enters the cell. When sodium enters the cell, what happens to the membrane potential? It depolarizes. When the membrane potential enters the cell, what happens to the voltage-gated sodium channels? They open. And when the voltage-gated sodium channels open, what happens to sodium? It comes into the cell. And when sodium comes into the cell, what happens to the resting membrane potential? It depolarizes. And when it depolarizes, what happens to the voltage-gated sodium channels? I think by now, you should have gotten my drift. What you get set up is a positive feedback cycle as shown in this diagram. Once you start the cycle, you have membrane depolarization. It opens the voltage-gated sodium channels. Sodium comes in, but the entry of sodium itself causes membrane potential, depolarization, which opens the voltage-gated sodium channels, which causes sodium to come in, which causes membrane depolarization, which opens the voltage-gated sodium channels, and so on and so on. And so we have a positive feedback cycle. The point at which the positive feedback cycle is set up is called threshold, because at this point, because of the positive feedback cycle, you get a very rapid and very fast opening of several thousand voltage-gated sodium channels. You get a massive influx of sodium into the cell, and that causes this rising phase, or phase 0, as sodium rushes into the cell, brings with it its positive charge, and therefore makes the inside of the cell more positive compared to the outside, and reverses the polarity. Now, I'm not going to explain that positive feedback cycle over and over again. But if you haven't understood what has happened there, what I want you to do right now is pause and just rewind the video a little bit and understand what's responsible for that rising phase and how it's generated due to the properties of the voltage-gated sodium channels and how you achieve this positive feedback cycle. So there aren't many places where positive feedback cycles exist in the body, but they tend to be dangerous because they produce rapid, large changes. But this is one time where a rapid, large change is needed, and it's used to generate the action potential. Now, the next question we want to ask ourselves is, well, why does phase 1 end? Why isn't there a continuous movement of sodium into the cell, and that this rising phase just gets higher and higher and higher and higher? Why does it always stop around plus 30 millivolts or plus 50 millivolts? Well, that's because these voltage-gated channels, they have certain properties. We know these from experimental studies, where scientists have been able to isolate one particular channel in what's called patch clamp experiments. When you isolate one channel, you can depolarize that channel, as shown on the top right here, by applying a current. And I want you to see what happens to the channel when you apply the current. The channel immediately opens, as we just told you and we explained to you. And it stays open for just a millisecond or so. But I want you to notice that even though the membrane is depolarized, after that millisecond or even less, the channel suddenly closes. Now, this is a unique property of these voltage-gated sodium channels, that they open in response to depolarization, but after a while they slam shut. A good analogy for you to think about is those of you who have electronic gates at home. Some of you know that if you have an electronic gate, when you press your remote control to open the gate will open, and it will stay open for a certain period of time, during which you can drive your car on the inside, or you can walk inside. But after a while, your gate is pre-programmed to close automatically on its own. Well, that's exactly how these channels work. When the membrane is depolarized, they open, but they're pre-programmed to close on their own. And I want you to notice something else here, that even though the membrane remains depolarized, notice the channel isn't open again. So we can say that these voltage-gated sodium channels go through three states. When the channel is at rest, it's usually closed, as we can see in this state here. The activation gate is closed. When the membrane is depolarized, the activation gate opens, and sodium is able to come through the channel. This is a very, very rapid process that happens in less than a millisecond. After a while, about a millisecond, a second process activates due to the inactivation gate here. and that closes. And when the inactivation gate closes, even though this activation gate is open, sodium cannot get inside. The channel is no longer functional. And this stays like this, regardless of whether the membrane is depolarized or not. After a few milliseconds, the inactivation gate is removed and the channel returns to its normal state. And so the sodium channels, the voltage-gated sodium channels, cycle through these three phases, where it's closed, but it can be activated, where it's activated and sodium is able to come in because of membrane depolarization, where it's inactivated, and then it recovers back into its closed but potentially activated state. Now, these three phases are responsible now for what stops the rising phase. Because after that 1 millisecond or so, all of the sodium channels begin to change. This means sodium can no longer enter the cell. And that positive charge is no longer entering the cell. So again, what I want you to do is, if you haven't understood what I've said, pause the video here, rewind it, and look at my description of how this voltage-gated sodium channel flows through these different stages. Now we're going back here to see that the rising phase stops because the voltage-gated sodium channels are closing. But something else important is happening, which is now going to trigger the repolarization phase, or phase 2. And that is the opening of voltage-gated potassium channels. These potassium channels also respond to voltage, just like the sodium channels, but they are very slow to activate. So you can see here from this diagram, at around -75, -50, they're only now starting to activate, and they slowly ramp up. So what happens is, as the voltage-gated sodium channels are closing, the slow activating potassium channels have now ramped up, and they're all now opening. Now, when the potassium channels open, what will happen? Well, remember, potassium is in a high concentration on the inside, and it's in a low concentration on the outside of the cell. So potassium is now going to leave the cell, and with it leaving the cell, it's going to cause positive charge to leave the cell. And the end result of that is you're going to have a return to the negative resting membrane potential. You're going to get phase 2 of our action potential, or the repolarization phase. Because these potassium channels stay open for a long time before they automatically inactivate, because these channels inactivate, there's not a gate that closes them. But as the membrane potential returns to normal, they inactivate. And because this is a much longer process, you find that the resting membrane potential, it actually ends up overshooting. As you can see here, it overshoots, and it goes a little lower into phase 3, or the hyperpolarization phase. But eventually, all of the potassium channels, these are voltage-gated potassium channels, they inactivate and close. No more potassium can enter the cell via these channels. Only the leaky potassium channels are open, and the resting membrane potential is reestablished. So what we can see here is it's the properties of the voltage-gated sodium channels and the voltage-gated potassium channels that are responsible for the generation of the action potential. And at different phases of the action potential, there's a different flux of currents due to different ions. You can see here in phase 1 of the action potential, the reason we have the rising phase is because of this very rapid influx of sodium. But that sodium influx is just as rapid, but it quickly stops as the voltage-gated sodium channel is closed and returns to 0. As the potassium channels open, you have this slow rising of the potassium conductance. But that stays open, these channels stay open a lot longer. And as they slowly close, you get a return to the resting membrane potential. And you get this repolarization, hyperpolarization phase, and a return to the RMP. So understanding the properties of these channels and understanding the ionic fluxes is what is responsible for the generation of these action potentials. Now, these things are very, very important, because if you want to stop an action potential, or if you want to block an action potential, the way you can do it is you can understand how these channels work. And what I would like you to do is I would like you to go and look up for a drug, a common drug that is used that blocks voltage-gated sodium channels. Blocks voltage-gated sodium channels. Now, if you block voltage-gated sodium channels, you cannot generate an action potential. And so that action potential cannot be utilized to communicate within the nervous system. So these drugs are very important when you want to manage pain. So I've given you a clue right there of where you should look to find out a type of drug. So please go and look for a drug that blocks voltage-gated sodium channels. And the other thing that I want you to do is I want you to go and I want you to look in nature, do some research online, because there are many toxins that are produced by animals and plants that interfere with the action potential by interfering with these channels. And they will give you a good understanding to appreciate better the generation of the action potential. Now let's talk about something called the refractory period. The refractory period. Now the refractory period is a period of time when you cannot generate another action potential, no matter what you do to the membrane potential. Remember, an action potential is usually generated when you depolarize the cell to threshold. In this example, threshold appears at about -55 millivolts. But the refractory period is when no matter how you depolarize the cell, you cannot generate another action potential. Now, there are two types of refractory periods. There's the absolute refractory period, shown here in a kind of pale orange color. And there's the relative refractory period, shown in blue. During the absolute refractory period, no matter what you do, you cannot generate another action potential. And this is because the sodium channels have gone into their inactivated state. The inactivation gate is closed. So you can depolarize that cell as much as you want, all the way up to 0 millivolts, up to 20 millivolts. You're not going to get another action potential. Those sodium channels are slammed shut, and they're going to be closed for a period of time. It's a very short period of time, just a millisecond or just over a millisecond. The relative refractory period, on the other hand, you can generate an action potential, but you need to apply a bigger current than usual to get to threshold. And this is because the cell is in a hyperpolarized state. So to get to threshold from, say, -80 here to get to -55, the value that you now have to apply is 25 millivolts, whereas before at the RMP, it was simply 15. So during the relative refractory period, you can generate an action potential, but you need to apply a larger current. And we're going to see the advantage of these refractory periods when it comes to the conduction of the action potential. So the refractory period is a period of time when it's impossible or more difficult to generate an action potential, even if we depolarize the membrane. There's the absolute refractory period where you cannot get any new action potentials being generated. And then there's a relative refractory period where you can get a second action potential, but you have to apply a larger depolarizing current. The final thing we want to talk about now is when an action potential is generated, how does it move along the nerve? One of the things you should have studied by now is the structure of a nerve cell. And a nerve cell, as you know, has a cell body. And close to the cell body is the axon hillock. The axon hillock is an area that has a high concentration of voltage-gated sodium channels. And usually the action potential is generated at the axon hillock. Then there's a long process in most nerves called the axon, the nerve axon, which leads to the terminal end of the nerve cell where you have the dendrites. So we want to talk about if an action potential is generated at the axon hillock, how does it spread? How does it move all the way down to the other end of the nerve cell along the axon? So let's look at this diagram here. Let's imagine this area in orange is the axon hillock. And we've just started the process by generating an action potential. So now the inside of this cell is positive in this region relative to the outside. The region next to it still has a normal RMP, with the region being negative. Now, when you have positive charge next to negative charge, what happens? They attract one another, and there's a flow of current, and the positive charge flows in this direction. But when positive charge comes here, what do we call that? Remember, when the inside of the cell becomes more positive, we call that a depolarization. And when that depolarization occurs, remember what happens to the voltage-gated sodium channels? They open. And when the voltage-gated sodium channels open, what happens to the sodium? It comes inside the cell. So once a positive charge flows over here and you get depolarization, you now open sodium channels and sodium comes in, and you get more depolarization, and more sodium channels open, and more sodium comes in, and you start the positive feedback cycle over here. And suddenly, you have an action potential being generated here. The action potential has regenerated and it's moved down the cell. Now, the question that we want to ask now is what's going to happen next? And I'm sure you can work it out. The positive charge is now going to flow to the next segment. It's going to cause depolarization. It's going to cause the opening of voltage-gated sodium channels. It's going to cause sodium to come in. And the action potential is now going to flow to this segment. And so the action potential moves down the cell along the axon by a process of regenerating itself each time. And for this reason, the action potential never gets smaller and is conducted without decrement, as we said earlier. So again, you can pause and you can replay my explanation. Now, one of the things you should have been paying attention to, or you should have noticed, is why don't we get an action potential moving in the opposite direction? Isn't positive charge also going to flow in this direction, backwards? Well, the reason for that not occurring is simply because, remember, this region of the axon would have just undergone an action potential. And the sodium channels would still be in their inactivated state. It would still be in the refractory state. And therefore, you cannot generate an action potential moving in this direction. So in normal biology, when the action potential starts at the axon hillock, the properties of the cell body stop it moving to the cell body. And once it starts moving down the axon, because of the refractory period, it can't move in both directions. So the direction of movement is unidirectional. Now, in a lab, you could stimulate a nerve in the middle of the axon, and you could get action potentials moving in both directions. But within the context of normal biology, within the brain and within the nervous system, the action potentials can only move in one direction. They're unidirectional because of the absolute and relative refractory periods. So I'll just run through this movement again. You can see it moving from segment to segment because of the regenerative nature of the action potentials. So this diagram here is simply another diagram that is explaining what I was just telling you. The action potential starts here at the axon hillock, and it moves down the axon. This region is depolarizing. It spreads to this region, and it doesn't spread backwards because the region before is repolarizing. And therefore, the direction of travel is unidirectional. Now, I want you to notice that this is a nerve, and you should see that there's a certain feature of this nerve. This particular nerve has no myelin. It has no myelin. Okay. Now, I want to talk to you a little bit about what changes the speed of conduction as it moves down a nerve. Now, there's something called the length constant. The length constant. The length constant is the distance over which a response decays at 37% of its size. 37% of its size. Now, that's just due to physics and different things that are calculated. And you can see that the length constant is given by the resistance of the neuron divided, right, is divided by the internal capacitance of that neuron. Okay, so you can see that equation there. The resistance of the neuron membrane divided by the internal neuronal resistance, not capacitance as I said before. And you take the square root of that. Okay. Now, the internal neuronal resistance is actually determined by the diameter of the neuron. Smaller diameter neurons have higher internal neuronal resistances. The end result of that, okay, the end result of that means that your length constant is going to be smaller. And so the action potential is going to travel more slowly. Let me go over that again. The length constant, we use it to understand what's the speed of conduction as action potentials move down the nerve. And it's been calculated in physics as the square root of the resistance of the neuronal membrane divided by the internal neuronal resistance. Okay. You don't need to understand that, but you can do some reading on that. What is one factor that affects internal neuronal resistance? Well, that factor is diameter. Because a nerve has a smaller diameter, just like a wire has a smaller diameter, that increases internal resistance. And that makes the length constant smaller. And so small diameter neurons conduct action potentials much more slowly than large diameter neurons. So neurons that have to take information from your spinal cord to your big toe tend to be large diameter neurons, wanting to transmit information very, very quickly. What can affect the resistance of the neuronal membrane? Well, the presence or absence of myelin can change the resistance of the neuronal membrane. Okay. Myelin can change the resistance of the neuronal membrane. And the presence of myelin increases the resistance. And so the presence of myelin increases the speed of conduction. But the way it does that is it causes there to be a jumping. Now, when you see myelin on the nerves, myelin, as you know, is a fatty sheath that wraps itself around the nerve. But it doesn't do it in a continuous fashion. There are gaps in the myelin called nodes of Ranvier. And what this increase in membrane resistance does is it means that there's no transmission of ions through the myelin, but there can be transmission through the nodes. So what happens is when an action potential is generated at one node, the positive charge, once it enters, flows to the next node. And here you have voltage-gated sodium channels. And the depolarization here causes an action potential to generate here. And then the next one flows over here, and you get an action potential here. And the next one flows over here, and you get an action potential here. So rather than a continuous stream as occurs in an unmyelinated neuron, you have a jumping effect. And this jumping effect is called saltatory conduction. And that leads to myelinated nerves being able to transmit action potentials much more quickly than unmyelinated nerves. Certain disease processes attack myelin and cause a degeneration of the myelin sheath. And what that leads to is slowing of conduction, and that can greatly affect the nervous system. One such disease is multiple sclerosis. Now, what we're going to do here is play a little video that shows you saltatory conduction in action. Okay, so we're going to play this little video, and hopefully you'll be able to appreciate saltatory conduction in a better way.
Saltatory conduction. Conduction through myelinated nerve fiber. Due to the myelination of neurons, action potentials may only occur at the nodes of Ranvier. Myelin is made up of insulating cells, which means depolarization cannot occur in myelinated regions. Between these cells, however, there are gaps known as nodes of Ranvier which are unmyelinated. As depolarization cannot occur at the cells making up the myelin sheath, the wave of depolarization can only occur at the nodes of Ranvier. Thus, action potentials appear to jump from node to node when traveling down an axon, and this phenomenon is known as saltatory conduction. And it serves as a means of increasing the rate of propagation of an action potential. Not only does saltatory conduction increase the speed of impulse transmission by causing the depolarization process to jump from one node to the next, it also conserves energy for the axon as depolarization only occurs at the nodes and not along the whole length of the nerve fiber as in unmyelinated fibers. This causes up to 100 times less movement of ions that would otherwise be necessary, therefore conserving the energy required to re-establish the sodium and potassium concentration differences across the membranes following a series of action potentials being propagated along the fiber.
So we've just watched that little video that hopefully has explained saltatory conduction and given you a good visual to help you understand what saltatory conduction is. So that brings us to an end of our study of action potentials. In this particular lecture, in this particular video, we've looked at a definition of an action potential. We've compared action potentials with local potentials. We've divided an action potential into 4 phases, phases 0, 1, 2, and 3. And we've looked at the ionic changes and the changes in the gates that are responsible for generating the action potential. We've spoken about the refractory periods of action potentials. And then finally, we've looked at the processes responsible for the movement of an action potential down a nerve. I hope you found this video useful, and I look forward to speaking to you again in another lecture soon.